Evelyn Fox Keller is professor of history and philosophy of science in the program in Science, Technology, and Society at the Massachusetts Institute of Technology. She is the recipient of a MacArthur Fellowship and holds numerous honorary degrees. She received her PhD in theoretical physics and actually did her dissertation in molecular biology at Harvard, which seemed like conflicting disciplines. Really an interesting combination of intellect that she has worked for years and years on the interface of physics and biology. She's authored numerous books, including A Feeling for the Organism, The Life and Work of Barbara McClintock, Reflections on Gender and Science, Secrets of Life, Secrets of Death, Essays on Language, Gender and Science, and many more. She lives in Cambridge, Massachusetts. She has 2 grown children, keeps a house on the edge of Cape Cod where she loves to garden and kayak in the cold Atlantic. So that's a little bit about her. It's my great privilege to introduce Evelyn Fox Keller.
Thanks very much. Well, thank you very much for that invitation, and I just want to say what a pleasure it is to be here in this wonderful city of Portland, which is widely known as the most livable city in America, right? But I have to say that I think the last time I was on a stage in a theater like this was when I was 8 years old, and my tap dancing school gave a performance in the Boulevard Theater in Queens, New York. Only then, I didn't have my name out in lights. So this is really quite exciting. Okay, so you've already heard that I'm a historian and philosopher of science. And my particular interest is in changes in the direction of scientific, especially biological research. I'm interested in how these changes come about, how they transform our questions, our understanding, and our expectations. Now, we seem to be living through some of the most cataclysmic changes in biology, so it's perhaps inevitable that over the last few years, I've turned my attention to the changes that are happening right now under our very feet. Of course, one faces pitfalls in trying to be a historian of the present, and probably the most serious of these pitfalls, especially in exciting times like ours, is that history can happen a lot faster than a scholar like me can write. So let me illustrate. Almost 5 years ago, I published a book called The Century of the Gene. This book was an attempt to map a trajectory of the gene concept from the time of the rediscovery of Mendel's laws in 1900 to the sequencing of the human genome. Well, the first draft came out in 2000, which made a wonderfully neat century. The book was both a celebration of the productivity of the concept of the gene throughout the century, and at the same time, a somewhat impassioned argument for the need to move on into what I'm here today calling the century beyond the gene. I agreed with William Gelbart, a molecular biologist at Harvard University, who wrote, "Unlike chromosomes, genes are not physical objects, but are merely concepts that have acquired a great deal of historical baggage over the past decades." And he went on to say, and we may well have come to the point where the use of the term gene might in fact be a hindrance to our understanding. Now, some molecular biologists read my book as anti-genetics, but this was a very serious mistake. My point was that if the 20th century was the century of the gene, the 21st would, in all likelihood, be the century of genetics, or rather, of genetic systems. The difference is very important, so let me explain. Genetics, I take to be the study of the processing of DNA in the construction of phenotype. of, of the organism. Genes, I take to refer to those entities that were historically assumed to be their particulate units of inheritance. The former I take to refer to the biochemical, that is genetics. I take to refer to the biochemical interactions underlying the construction of actual organisms. And the latter, genes, I take to refer to a hypothetical conceptual scheme. Now, the principal historical baggage of the gene concept dates back to the view of genes as the basic units, that is, the atoms of life. But what is a gene? The fact of the matter is that molecular biologists today employ a number of different definitions, and they need all these variations. On one definition, a gene is a specified stretch of DNA, passed on intact from generation to generation. But which stretches of DNA count as a gene? Those that code for proteins? Or should we include those that code for bits of RNA that are crucial for regulation but are never translated into proteins? Or many other areas or sections of DNA sequence are crucial for the functioning of the organism. We don't usually call them genes. Or on another definition, a gene is the coding sequence corresponding to a particular protein, a sequence that for higher organisms exists as such only in the form of messenger RNA after it has been processed. Let me explain. What happens in the cell is that there's an elaborate machinery for transcribing the DNA or parts of the DNA onto messenger RNA, and that messenger RNA is not by itself, that raw messenger RNA is not useful in the process of protein synthesis. Before we can proceed to the step of protein synthesis, that long stretch of messenger RNA needs to be edited, it needs to be spliced, it needs to be pieced together to correspond to a particular coding sequence. Only by splicing together these, sometimes the segments can be shuffled, just like we do with our word processor. But only by splicing together these shuffled segments of the original DNA sequences can a molecule corresponding to the protein in question be constructed. This molecule that corresponds to the protein might be said to have existed as a chromosomal entity only in potentia. Okay, even more problematic are those proteins that are constructed from messenger RNA transcripts that have been specifically modified at particular developmental stages, long after they have been transcribed. For example, there are molecular machines that will attach themselves to the messenger RNA and snip out one nucleotide and replace it by another. Or just insert several other nucleotides that don't correspond to anything on the DNA. So when we ask how many genes are on the human genome, and that was when the first draft of the human genome was announced, that was a cause of great deal of consternation, that there were only 25,000 genes on the Human Genome Project. What does that number mean? That was very disturbing to people because we should have many more genes than, say, a fruit fly or a worm. But what does that mean? When we ask how many genes are on the human genome, the answer we get will vary according to which definition of gene one employs. And the answers will differ. For example, if you use the definition of a gene that corresponds to a particular protein, you might get a number that's 2 or 3 orders of magnitude greater than the number of genes defined as stretches of the DNA. So given all the difficulties of gene counting, some people suggest that it might be better to give up the whole idea of counting genes, or even perhaps even referring to genes, and concentrate on particular molecular parts, a parts list that are based on functional protein domains. Now, by contrast to the gene, we do know what DNA is. It's a very specific molecule. We can spell out its sequence, and we can observe the remarkable stability of that sequence over the course of the generations. That is to say, there are sometimes mutations that take place in the transmission of the DNA pattern from one generation to another, but remarkably few mutations. But the most important lesson we have learned is that virtually every biologically significant property that is conventionally attributed to the DNA, including its stability, is in fact a relational property. It is a consequence, the stability, for example, of the DNA is a consequence of the dynamic interactions between DNA and the many protein processors that converge upon it. Indeed, the very meaning of a DNA sequence is relational. In other words, one could say, if you take a stretch of DNA by itself, just naked DNA, and ask, are there genes on that stretch of DNA? And the answer is no. Genes do not exist on naked DNA. Genes are meaningful units, the meaning of which is established only in relation to the processes, all that molecular machinery that's involved in interpreting the DNA and translating it, and et cetera, et cetera. For the purposes of understanding development or disease, furthermore, it's not even just the sequence that we need to know, but for the purposes of understanding development and disease, it's the patterns of gene expression that matter. And these patterns are under the control of an even more complex regulatory apparatus, and the patterns of gene expression, make sure you get it, you understand this, the patterns of gene expression cannot be predicted from knowledge of the sequence alone. So, this is what I argued in the book. I probably should have waited. 5 years ago, the number of molecular geneticists willing to give up on their paradigm of genetic reductionism was still relatively small. But today, biologists seem to be undergoing a paradigm shift right under our noses. Overnight, as it were, biology departments have begun to jump on the bandwagon that's called systems biology. Now, in my book, I gave abundant thanks to the Human Genome Initiative for this shift, somewhat ironically, since I had been an early critic of the Human Genome Initiative, but now I credit that project with restoring our humility. But it may well be that I was premature. I may have jumped the gun. But it is clear today it's clear that it has, I apologize. The problem with editing your script before you go is that you're not very consistent. At least I'm not. So it is clear that this shift has taken place today, and it also seems that my thanks to the human genome were well placed. A few quotes will make the case for this. From the Bauer Center at Harvard University, we read, quote, "The completion of the draft sequence of the human genome has marked a turning point in molecular biology and has stimulated a broad movement towards systems-wide approaches to biological complexity. The central question has shifted from who are the actors to what are the scripts? How does collective behavior emerge from the actions of individual actors? Or take a lead article from a recent issue of The Scientist, where it is written, "For 50 years, biologists have focused on reducing life to its constituent parts, first focusing on the cell, then working their way down to the genome itself. However, such achievements created a new challenge, making sense of the huge amounts of data produced. As Professor Dennis Noble from Oxford University puts it, it took Humpty Dumpty apart, but left the challenge of putting him back together again." Close quote. In a similar vein, at the U.S. Department of Energy, where the project of sequencing the human genome was first born, a new project is now underway. It is called Bringing Genomes to Life Program, and it is explained as follows. From the foundation of whole genome sequences, the aspiration of the new biology is to build a new, comprehensive, and profound understanding of complex living systems. The current paradigm in biology, variously described as single gene, reductionist, or linear, is not likely to be successful on its own. And then they go on, knowing the functions of all the genes in the genome by itself will not lead to understanding the processes of a living organism. Instead, they continue, the existing research approaches will be augmented by a systems approach. The new paradigm grows out of rapid advances in instrumentation for the biosciences, the vast improvements in computing speeds and modeling capabilities, the growing interest from physical and information scientists in biological problems, and the recognition that new approaches are needed for biology to achieve its full promise. Close quote. In other words, having helped take the genome apart, the Department of Energy is now embarked not only on putting it back together again, but also in bringing it to life. Somewhere along the line, people have come to realize that the genome by itself is not, in fact, alive. Somewhere along the line, we seem to have learned that vitality, as Linus Pauling long ago reminded us, does not reside in the molecules, but in the relations among them. The architects of the new DOE program write, "We need to figure out what these parts do in relation to each other." In short, they are telling us that we need a systems biology. In much the same spirit, MIT, a couple years ago, had launched a new computational and systems biology initiative. And the mission of this new initiative was to, quote, "to lay the foundation for treating biological entities as complex living systems rather than as an amalgam of individual molecules." In his opening address at the first annual conference of this initiative, then president of MIT, Charles Vest, said, "Until now, biologists have learned more and more about the detailed structure and functions of the molecular components of life. But we have not yet understood how individual components are networked to control physiology. Now we are in a position to begin the search to understand our molecular machines and cell circuitry, how the parts are connected, how they operate. In a third revolutionary transformation, biology will become a systems science." Close quote. Let me show you a couple of slides that were taken from that conference to give you a bit of the spirit. First, it begins with I'll hold it. Okay. The parts list is not enough. Well, here they estimate the number of genes. This is coming from the first draft of the human genome. It's 30,000. That estimate has gone down since. But again, I have to remind you that the meaning of that number is very questionable. And the observation that the mouse genome has the same number of genes. How can that be? What makes a man and a mouse a mouse? Well, obviously it's not in the number of genes, nor is it even in the kind of genes, because many of those genes, a surprising number, are practically the same. What makes a man and a mouse is the relations between those genes, the way they are hooked up to each other, the way they are regulated. Everything depends on regulation. Or here's an example of a regulatory system, a biological system, and the point that you can read it, that the regulation of biological functions needs to be understood as complex biomolecular circuits. Or just one more. Now, there are a lot of engineers there, and engineers know quite a lot about circuits, right? So the emphasis was on the parallel, on the similarity between electronic circuits. Doesn't look so similar here, but the logical wiring of the electronic circuits matches the logical wiring of the biological circuits. Okay. I could go on. Efforts of a similar kind are gathering strength all over the US and Europe. Harvard Medical School has opened an entire new department called systems biology. Now, two questions jump out at us right away. The first is, what drives this move to systems biology? Why now? And second, what does it mean? What is systems biology? The first question is relatively easy to answer, the second a bit more difficult, so let me start with the first. Why now? The most important impetus seems clearly to be the realization of the Human Genome Initiative's first set of goals. It's already evident from the quotes I have read to you that there's a widespread sense that the reductionist phase of genetic research is now over. Stephen Benner speaks for many when he writes, "Sequencing the human genome represents a culmination of sorts of chemical reductionism, and now we need to move on." But also, the completion of the first phase of the Human Genome Initiative brought with it something of a disappointment. The human genome has been sequenced, but it has failed to tell us who we are. As the Department of Energy puts it, we now have the parts list for these organisms, and we can see that knowing the parts, even knowing the function of the parts, is not enough. But the Human Genome Initiative and molecular genetics more generally have given us far more than a parts list over the past decades, and this more than the parts list really needs to be credited and acknowledged. They have also given us new tools for going beyond the individual parts. They've given us tools for probing the complexity of cellular dynamics, and together, the tools and this parts list have produced a veritable avalanche of data. Just a few years ago, Sidney Brenner wrote, "There seems to be no limit to the amount of information that we can accumulate, and today, at the end of the millennium, we face the question of what is to be done with all this information." It has become evident to people, that's the end of Brenner's quote, it has become evident that the methods of data analysis and interpretation that have traditionally been available to molecular biologists no longer suffice, and that new methods are required, perhaps even to be borrowed from other disciplines. But as it happens, fortunately enough, technical developments in computer science, together with the recent emergence of an entire discipline of computational analysis designed precisely for dealing with massive amounts of complex data, have guaranteed that such new methods are available. Furthermore, their integration into molecular biology, conjoined with the techniques of molecular analysis, for example, gene chips, has proven not only necessary, but enormously productive. In fact, the influx of huge numbers of physicists and engineers into molecular biology today is one of the most conspicuous marks of the new systems biology. But that takes me to the next question. What is systems biology? In brief, I want to argue that so far, systems biology is a concept or a term waiting for definition. The term itself invokes the name of Ludwig von Bertalanffy from the earlier part of the century and his strong advocacy of a systems biology beginning in the 1930s. With somewhat uncharacteristic modesty, von Bertalanffy wrote, "The notion of a system may be seen as simply a more self-conscious and generic term for the dynamic interrelatedness of components." Close quote. Today, however, advocates for a systems biology have much more to work with than von Bertalanffy did, and they are accordingly less modest. They want, both literally and figuratively, to put Humpty Dumpty back together again, and they have great hopes for the gains they expect to follow. President Vest at MIT voiced particularly high hopes in his address to the new Computational and Systems Biology Initiative. Quote, "The ability to better understand and predict the actions of complex biological systems is expected to lead to advances in drug design, disease diagnosis, biologically inspired computers, environmental health, and national defense." What more could you want? Of course, we can recognize this as hype, and it's a hype of a kind that is precisely Vest's job as president of a large institution like MIT. But Vest is far from alone, and his comments echo the sentiments of many others. Leroy Hood, founder of Systems Biology Institute in Seattle, explains, "Unlike traditional biology that has examined single genes or proteins in isolation, systems biology simultaneously studies the complex interaction of many levels of biological information: genomic DNA, messenger RNA, proteins, functional proteins, information pathways, information networks. They study this in order to understand how all these parts work together. They're not even parts, all these processes work together." And almost invariably, especially for engineering scientists, understanding how the parts work together is followed for understanding, as it was not for Bertalanffy, for, quote, "understanding how to control the system and how to design the system." In other words, as the term systems biology is now used, it is an exceedingly plastic and multifaceted concept. It refers to a conceptual analysis, it refers to a technological agenda. Synthetic biology has become a large field now under the rubric of systems biology. It calls on a wide range of skills and promises an equally wide range of results. It is a siren call to scientists from disciplines far afield from biology, from engineering, from computer science, from physics, from mathematics. These are all disciplines for which experimental biology has in the past had remarkably little use. Moreover, they are disciplines where the lack of a common theoretical framework is conspicuous. One might even say it's a little bit reminiscent of Pirandello's 6 characters in search of an author. Nevertheless, there is today an unprecedented demand for the skills of computational, mathematical, and physical scientists. But there is something qualitatively new in this reincarnation of a mathematical or computational biology. At least so far, and probably for the first time in history, the invitation is coming from out of biology. It's not like it has been in the past, physicists looking for new areas of employment or new fields to plow. Now, biologists are setting the demand in the sense that they are simply asking the new recruits for help in integrating and making functional sense of the data that came from their recent triumphs. This may change, but for now at least in most places, the biologists seem to be in the driver's seat. Now, recognition of the demand for new tools of analysis in molecular biology created by the deluge of data now pouring in was not, however, the main point of Sidney Brenner's 1999 paper that I cited just a few minutes ago. Brenner went on to argue that the prime intellectual task of the future lies in constructing an appropriate theoretical framework for biology. Unquote. And this leads to ask another question. What would an appropriate theoretical framework look like? Does systems biology provide us with such a framework? Well, you already know that from my remarks that I've made, I don't believe that it does. There are too many different fields involved, too many different conceptions of a theoretical framework. They're all working in parallel, it seems. From where, then, might such a framework be expected to come? And what would it look like? I have no crystal ball to help me answer this question, but there are some obvious candidates out there, and many of you will come to your mind almost immediately. So perhaps I can make use of some history to cast light on the future and offer a few comments about the likely promise of these obvious candidates. One such candidate, and probably the most obvious, comes from physics, the discipline that for more than 3 centuries has been accepted by almost all philosophers of science, by almost all philosophers, and by almost all of us, has been accepted as the traditional arbiter of theory in the natural sciences. Indeed, for many scientists, the words theory and physics are virtually synonymous. And so it is that at a recent meeting outside Aspen, Colorado, several dozen theoretical physicists with an interest in biology gathered to celebrate. And here I quote from a report in Nature. They gathered to celebrate the, quote, the growing feeling that their discipline's mindset will be crucial to reaping the harvest of biology's post-genomic era. Close quote. And this is not only in Aspen, but all around the country, even around the world. Physicists and mathematicians have heard the siren call, and they are accordingly looking to the life sciences for new fields to ply their trade. That is to say, there are many physicists out there who are responding to the new siren call in exactly the same terms as they have in past decades. They are looking to the life scientists with what one reporter calls bioenvy. Some say that physicists are looking for ways to reclaim lost glory. Others suggest a more pragmatic goal, that biology is where the money now is. Or, somewhat more high-mindedly, one might say, here is where the intellectual and scientific action now is. If the last century belonged to physics, the new century, it is frequently said, belongs to biology, and I think that is right. Observers also note, however, that the move from physics to biology is not quite so simple, that there's a certain culture gap between the disciplines that needs bridging, that biologists and physicists have different goals, different traditions, and they ask different kinds of questions. They even look for different kinds of answers. If that gap is to be bridged, some resolution of or some accommodation to these differences is required. Indeed, as I've already said, this is not the first time that physicists and mathematicians have turned to biology for new problems, new fields to plow. But the history of such efforts is rather dismal, at least from the perspective of building bridges between the two cultures. And biologists have shown very little interest in past efforts to mathematize or theorize their discipline. More typically, they have shown impatience and even indignation. Apparently, this is no longer the case. Today, collaborations between experimental biologists, physicists, and mathematicians are everywhere. Indeed, the place of mathematics in biology now seems to have entered an entirely new phase. Over the last few years, both the NSF and the NIH, but especially the NSF, have launched a number of large-scale initiatives aimed specifically at realizing the potential of mathematical and computational approaches in biology. Since 1983, just 20 years, the proportion of funding for mathematical and computational research coming from the biological division of the NSF has increased 50-fold. That's just proportions I'm measuring, not absolute quantities. Also contributing to the new resources are a number of private foundations, like the Keck Initiative for exploring new directions for interdisciplinary collaborations. Indeed, the complaint that is most frequently heard today is one of a paucity of applicants rather than a paucity of funding. This increase in resources is in turn reflected in a corresponding growth both in the rate of publications and the formation of new programs in mathematical and computational biology that we've been talking about. And unlike their precursors, these programs are now housed in departments of experimental biology and not, as they were in the past, in mathematics or computer science departments. Furthermore, the new recruits coming from the mathematical sciences do not simply collaborate with experimental biologists. Many of them often become practicing biologists themselves. Conversely, biologists today no longer need to simply hand over their questions and data to others, thanks in part to the rise of computer literacy and in part to the development of user-friendly computer programs that bring the techniques of mathematical analysis within the grasp of those with little or no conventional training in the subject. Now they can build models of their own. The net effect is the beginning of an entirely new culture that is simultaneously theoretical and experimental. A culture that doesn't tolerate any splitting, as physics has historically done, into theoretical and experimental science. It gives rise to a breed of mathematician biologists that is as familiar with handling differential equations as with the limitations of messy experimental data. If, as a number of people now claim, and as the evidence supports, a new mathematical biology is emerging, it is because the beginnings of a reconciliation of the traditional cultural conflicts is in the air. Excuse me. The new mathematical biology brings with it not only new skills, but also new values. It gives rise to a discipline that bears only passing resemblance to the past efforts to do for biology what mathematics had done for physics. Efforts to make biology a theoretical science like that of physics. It promises not so much a vindication of past failures as a transformation of the methods, the aims, and the epistemological grounding of past efforts. So let me just list what I think are the key features of this transformation. And it's actually very exciting because it is transforming all of our ideas as we bring these two cultures together. Maybe we can envision some similar productivity on the political scene of bringing different cultures together. But just conceptually. So first, the most successful efforts at modeling suggest the need to completely rethink the meaning of many of our basic vocabulary, like essential or fundamental. No longer can we take the essence of a process to be found, can we assume it to be found in abstract or simple laws, but rather the essence of a process now seems to be found in the messy specificity of particular adaptations that have come into existence by the haphazard processes of evolution. It is the accidental particularities of biological structure that is what is fundamental, like that of DNA, for example. Fundamental now in a completely different sense than it has meant in the past, fundamental in the sense of having been built in historically on the ground floor, and hence as having become most deeply entrenched. In other words, if physicists are to be helpful in forging an appropriate theoretical framework, they will need to rethink some of their most basic epistemological assumptions. They will also need to rethink some of their traditional arrogance. So just by way of example, here are some quotes from the recent literature from physicists. Biology today is, this should be like a test. What is wrong with this picture? Okay. Biology today is where physics was at the beginning of the 20th century, close quote. Or, biology is faced with a lot of facts that need an explanation, close quote. Or, physics gives us deeper understanding. It can offer biology fundamental explanations. The fact of the matter is that biology has now begun to throw a very serious monkey wrench into all of our traditional assumptions about what ought to count as deep, as fundamental, about what counts as an explanation, or even about what we will count as progress. So here's another feature of the transformation I see happening. Biological systems are, as we know, extraordinarily complex. But again, because of evolution, they are complex in rather different ways than systems in physics are understood to be complex. For one, they are always and inevitably hierarchical. Accordingly, familiar terms, concepts like emergence, we're all very big on emergence, and the application of emergence to biology is, the talk of that is everywhere. And we all know about nonlinear dynamical systems and how that's going to teach us about emergence. But those familiar notions of emergence that are rooted in nonlinear dynamics are rooted in the nonlinear dynamics of uniform systems. That's what we have gotten from physics. The understanding of the enormously interesting effects that come out of the dynamics of uniform gases, fluids, or lattices. But such concepts are not adequate to the task. What is different about the reality of biological systems is that the huge number of functionally diverse components, the enormous heterogeneity of the system, and the frequently multifunctional sets of elements that interact selectively and nonlinearly to produce coherent rather than complex behavior. So maybe complexity is a word that has been worn too thin. Maybe we need to have a new term to characterize what is distinctive about the biological system. The central point is that the inhomogeneities and ordered particularities of biological systems are essential to their functioning and cannot be ignored. Indeed, to ignore them is to risk exactly the kind of biological irrelevance that has historically been the fate of so many mathematical models in biology. The third point. Because of the character of biological complexity, useful models of biological systems tend not to be mathematical in the usual sense. They are computational. Nor are these models stepping stones to a final theory. The models themselves are the theory. Or to put it somewhat differently, theoretical biology will not be formulated in a few simple differential equations. Rather, what we will recognize as theoretical biology will be a messy complex of algorithms, of vast systems of differential equations, statistical analyses, simulations, and throw in whatever else you can think of too. Such models can only be successfully formulated in the most intimate back and forth relation with experiment. And in fact, I sometimes think that the best use of the term model in biology is a verb, as a verb, but verbs are going to recur in my comments. Finally, distinctions between pure and applied. That's another distinction that we're all familiar with, pure science versus applied science, theoretical science versus practical science. These are distinctions that are absolutely basic to our contemporary views of science, and especially to physics. But these distinctions also have to go. And in large part because of the very technology that has enabled the collection of so much data in biology. Techniques of recombinant DNA have made it possible to directly intervene in the internal dynamics of development. They have turned genetic markers into handles for affecting specific kinds of change. In short, the very technology that has paved the way for theoretical biology has also and simultaneously made genetic engineering a reality. And, it should be said, also a business. Biology is becoming a practical science in the very same move as it turns theoretical. In fact, in biology, the two terms, practical and theoretical, have become almost impossible to disentangle. Okay, I mentioned two candidates for a possible theoretical framework. The second, and in many ways more promising, the second and in many ways more promising candidate for a new theoretical framework comes from computer science and from our current love affair with the image of the organism as computer, and for many of biology as a digital science. Now, I say this is more promising precisely because of the rich practical tools computer science has given us for thinking about interactive systems. In many ways, these are tools and metaphors that take us far closer to the complexity of biological systems than do the traditional models of theoretical physics. But there's surely a serious error in overemphasizing the digital aspects of genetic processing and overlooking the fundamentally analog nature of the chemistry that underlies all such processing. Indeed, we have been taught to think of DNA as a digital string, of the sequence as bytes. It's information encoded in the sequence of nucleotides. But in fact, real-world DNA, functional DNA, is also a three-dimensional molecule. And the way that three-dimensional molecule is wrapped and twisted and contorted and shaped has an enormous influence on the expression of genetic information. So all that information, the shape of the DNA, the configuration, the twisting, that's all analog information. Furthermore, here too, there's a lesson to be learned from history. As scientists, our way to think about phenomena we do not understand is, as it always has been, and as it of necessity must be, is to liken the unfamiliar to the familiar. Thus, the image of the organism as a machine goes back to ancient theorizing about the nature of life. The only thing that has changed is what it is we think of as a machine. We started with machines as pulleys, hydropumps, clocks. We went on to steam engines. And we have displayed extraordinary ingenuity in constructing ever more versatile and more inspiring machines. Likening the organism to each of these machines has been very instructive in the past, just as likening the organism to our newest machines is instructive today. But it would hardly be reasonable to suppose that our ingenuity has run out, that we will not yet build even more lifelike machines. In fact, our best computer scientists are betting on our ability to do so, and once again, they're looking to biology for inspiration. So let's embrace the terms and the images of computer science to help us think about the systemic properties of cells and organisms, but let's not forget that these biological systems still have quite a few tricks to teach our engineers. Many people argue that systems biology would do better by looking to the engineering sciences than to the physical sciences for kinship, and perhaps that is so. But let's not forget that our engineers have yet to build a system that is both self-designing and self-generating, a system, in other words, that we would be willing to call alive. I'm not saying that such a task is in principle impossible, only emphasizing that our current machines, our current computers, our latest airplanes, and our most sophisticated internet systems are not up to the task. So what will it take to bring the genome to life, to formulate an appropriate theoretical framework for understanding living systems? This question, of course, I can't answer, but I want to hazard 3 suggestions. First, if I had to lay odds, I'd pay close attention to where the current limits of both engineering design and genomic science are now. And I'd ask, alongside our most advanced engineering scientists, have we put enough time in to enliven these systems? Have we taken a sufficient account of the temporal dynamics of our systems? Have we paid enough attention to the timekeeping of our different regulatory systems? Is it enough, for example, to think of genes as turning on and off, or must geneticists, like their colleagues in neuroscience, begin to examine the precise timing of these on-off switches, both relative to each other and to the temporal dynamics of global processes in the cell? Must we start thinking about all the interrelation of all these different temporal scales, these temporal processes, and the kinds of synchronization that Stephen Strogatz calls synchronization? He calls it self-organization in time. I think I'd put my money there for our next leap forward. But even if I'm right, attention to temporal dynamics does not in itself constitute an appropriate theoretical framework. Which leads me to my second suggestion, which comes out of the question, does systems biology in fact need a single coherent theoretical framework? Perhaps it can forge an adequate or at least workable scaffolding by molding, transforming, and combining elements of the theoretical traditions that have preceded it. In the absence of an available framework that can be imported from an already existing discipline, that may well be how things will proceed, for good or for bad. And based on an examination of the kinds of work that are thus far emerging under the name systems biology, that clearly seems to be the current modus vivendi. But many, even those who disavow the possibility of a single coherent theoretical framework, many are unhappy with this state of affairs, and they just wish for a bit more coherence than now seems available. Which takes me to my third and last suggestion. To the extent that our conceptual frameworks are constrained by lexicons, by languages that have been fashioned for other kinds of challenges, it may well be worth stepping back and taking a brief look at the linguistic habits that have underlain our existing theoretical traditions. Indeed, I suggest that the challenges now posed by our most recent encounter with biological complexity may require some new ways of talking. Most biologists may now agree on the need to shift their focus to the interaction between and among individual parts, and even to the dynamics of these interactions, but I suggest that in this effort they are handicapped by ingrained habits of thought and habits of speech that give ontological priority to the parts. When, for example, the architects of the new Department of Energy program that has been designed to bring the genome to life write, "We need to figure out what these parts do in relation to each other," the implicit assumption is that the parts come first, and only later, out of the interactions between and among those parts, do larger entities emerge. There are some obvious problems with this assumption, one of which is especially conspicuous in genetics, where the parts are taken to be genes. The problem is here, as I've already said, that genes do not have however we want to define the genes, whether we mean a segment of the DNA or the messenger RNA, however we define the gene, that term has no meaning in isolation. A sequence of nucleotides is constituted as a gene only in the presence of transcriptional and translational systems of the kind we find in a living cell. The cell, in other words, is a meaning-making system that turns nucleotide sequences into genes, again, however we define them. How then can it possibly be appropriate to think of the cell as constituted by genes and their products? What I'm suggesting is that prior to the need to construct an appropriate theoretical framework may well be the need to construct a more appropriate linguistic framework in which those interactions constitutes the parts themselves. The beginnings of a new lexicon is already evident as geneticists seek to forge new ways of thinking about biological function, as they look for clues to that function, not in the particular genes, nor in the structure of DNA or the protein products, but rather in the communication networks of which DNA and proteins are part. Of course, and I would never deny this, DNA sequences remain an absolutely critical resource in these efforts, both for the researcher as for the cell. But we are beginning to see a shift in focus in the search for biological function to the crosstalk between, among all the players of the cellular orchestra. Communication has become a new buzzword for biology. And here's a piece of a new lexicon. And it captures the discovery by traditionally reductionist life scientists of the power of sociality. This is a definite good, but communication is just one term. The more we learn about how the parts work, not only in interaction with each other, but also with the larger entities in which they are embedded, the more we learn about the extraordinarily complex and versatile systems of gene regulation, about the signals mediating all the different levels of organization, about the variety of epigenetic mechanisms of inheritance at play, and about the evolutionary feedback between all these different mechanisms, the more compelling is the need for an entirely new lexicon, one that has the capacity for representing the dynamic interactivity of living systems, and for describing the kinds of inherently relational entities that can emerge from these dynamics. As I said, time is crucial here. Time is the medium in which interactions occur. For too long, we have been trying to downplay, if not eliminate, temporality from our science, focusing on things, on entities, on the atoms of life, on the atoms of physics. In biology, these atoms are genes rather than, perhaps it was time to turn our attention to the processes. So here is a bold suggestion. Genes may well have outlived their usefulness as nouns, but perhaps they can be revivified for the 21st century as verbs. I think I'll stop there. So thank you all. I know that you have microphones set up for questions, and I'm here we've got lots of time to take whatever questions you have. So line up or pass in your questions on cards.
Is this Mike? Yeah. I want to correct one little oversight that I did, and that is to thank Mentor Graphics Foundation, which is different from Mentor Graphics Corporation, for a lot of tickets for the K-12. They're wonderful, and we love them, and a lot of you guys are here because of that. So there you go. And can we have the house lights up a little bit during the Q&A? If those guys are awake up there. And there's probably, is there someone over at the mic over here?
Yeah, let's have the house lights on.
Hi, Dr. Keller. I have a question for you.
I can't see. Okay.
You presented as if biology and physics were different or sort of at war with each other. Maybe that's an exaggeration. But I'm wondering if we heard from Brian Greene earlier, and the idea of string theory, I think, may bring a convergence between theoretical physics and biology, and as well as perhaps a convergence between Newtonian physics and quantum physics. So that's just an idea I have.
Okay, no, that's an interesting question. But maybe we can wait for just 1 minute or 1 or 2 minutes while people leave and the noise quiets down. What? That's better. Okay, good. Okay, so the question is, I've emphasized in my talk the differences between physics and biology. And the question is, but is there not a convergence between physics and biology taking place not only in the kinds of convergence I was talking about in the development of a new kind of mathematical biologist, but also theoretically in quantum mechanics, relativity? Actually, there is. It's a really interesting phenomenon. There's a quote that I often use, and I have used twice now, a quote from the physicist Paul Davies, when he talks about complexity, complex systems, the physics of complexity, and he talks about emergence and self-organized criticality and the processes that give rise to clouds or to waves or to turbulence or to snowflakes or to umbrellas or people. The original quote is much better. Now, this is one attempt to bring biology and physics back together again. It is an attempt that brings biology under the wing of physics. It erases the distinctions between people, umbrellas, and clouds. And I think that is, that is not going to give us an account of biology. Biology is not like clouds. Biological systems are inherently particular, specific, heterogeneous.
But the shape of string theory that he was talking about, this enfolded, multidimensional, multi-hold shape, is very biological, really. The way there's many dimensions within, and that alternating dimensions have the coding for the larger dimensions. Implied, so there's an implicate and an explicate nature and a relationship between these small, tiny, the tiniest things to the larger and to the largest of everything. And that's the part that seems very biological to me, too, and the enfolded nature, too, of the shape he describes.
Right. There are a lot of biological-like metaphors there, but you wouldn't want to call the dimensions of space alive. You wouldn't want to attribute vitality to them, would you?
It's interesting that a lot of the physicists that are working in terms of string theory have become very familiar with Buddhists and that idea of nested, enfolded consciousness. And I think there's, some possibilities that are beginning to emerge that are very interesting.
Oh, I agree with you. I agree with you entirely. The kinds of convergence we're having from different cultures and different perspectives are tremendously interesting. But they are interesting and they need to be looked at. So when I focus on biology and physics, and we talk about the convergence, I want to look carefully at the terms of that convergence. And I'm very struck by the fact, something you did not mention, but you might have, one of the expressions of convergence comes in cosmology. When Lee Smolin has a book called The Life of the Cosmos. And it's about, not only does he invoke the concept of a self-organizing system to refer both to biological organisms and the cosmos, but also he talks about birth and death. He talks about evolution, about natural selection, and I think this is very interesting. But I also want to know what are these metaphors doing for him? We mustn't forget that physics had no need for biology when it was, king of the heap, top of the heap. But
it was only looking at the very, very small and the very, very large. It never looked at the medium scale, the scale that we look at. And if all the scales are described by a unified description, a unified field theory, our holy grail of theoretical physicists, maybe it will describe biology as well, and cosmology.
Well, maybe it will. I personally think it won't. I personally think that the assumption that there is a unified theory that will accommodate all aspects of our experiential world, it needs to be seriously re-examined. But that's my personal view of these things. Let's take another. Okay.
Yes, so thank you, Dr. Keller, for your lecture. I'm probably a little different than very many people here. I'm 82 years old. I'm a full-time farmer at the time, but I read a dozen or more of the top science magazines of the world since I was a 10-year-old or 12-year-old boy in country grade school. And I'm really fascinated with genetics. There's a genetic book that I took from and read from the Oregon State University Library by the name of a fellow by the name of Reidi, R-E-I-D-I, and he says all of biology is genetics, and it was kind of a last-minute comment on his opening page. And I have a question. You were using the term molecular biology, and I was wondering how that is used in genetics.
Okay. When I talked about molecular biology, I should have said molecular genetics. Now, the question is all biology genetics? Well, partly, the answer to that is a definite no. There are so many processes, aspects of our functioning, and even of our development that do not involve the level of genetics. Even with my expanded definition of genetics, I define genetics as the processing of DNA. Okay, but there are a lot of aspects of biological organization that don't involve the DNA in any way. So biology is not all genetics.
Well, I appreciate your answer, Dr. Keller.
Thank you. I can't see. That's a good thing.
Yeah, right here. I was wondering if you think that changes in direction of scientific direction is fueled by breakthroughs in new technology, or is it fueled by I want to know this now instead of something like how you said physics was in the last century and genetics is the new century. I was wondering what that's fueled by. Is it technology?
That's a great question. I think that there are many motors of scientific inquiry, and which motors are dominant is there's no general answer to that. Let's look at the kinds of motors of scientific inquiry. There is economics. There's the marketplace. There is one of the great, very important motors for the development of physics in the middle of the 20th century was national defense. That enormous resources were poured into the development of physics, into training physicists, because it was argued that we need physics for national defense. Another motive for scientific inquiry is our innate curiosity, our need to understand, to make sense of the world. Another motive of scientific inquiry is greed, fame. There are a zillion different kinds of motives of scientific inquiry. Now, the emphasis in physics on national defense was extremely important in the middle of the century. It's actually been important historically. It's always been one of the main motors of the development of physics, but not in the early part of the century so much. So relativity and quantum mechanics did not come out of a need for national defense. What spurred those developments? Well, I actually think those developments were spurred by more intellectual, conceptual drives. What is spurring biology today? Well, what I didn't say, when I talked about the need for an appropriate theoretical framework, well, I raised the question, do we need a single theoretical framework? Do we need a unified framework? What I didn't raise is the question, do we need any kind of theoretical framework? A lot of biologists today don't feel the need for a theoretical framework of any kind, because biology is today, in a way that is quite worrisome, is driven by economic forces, by the market. That's a very serious phenomenon, and in many ways it's a new phenomenon. We need to look seriously at it. It's particularly a concern in biomedicine, and there's been a lot written in the last few years about the pharmaceutical industry and the way in which it is shaping and driving the research enterprise of biomedicine. So there are many. I can't give you a single answer.
Okay, thank you for your time.
Here's a question. I was sure somebody would ask this question about Larry Summers. You all know about Larry Summers. Is there anybody who wants an explanation of who Larry Summers is? Okay. Larry Summers, this is such a great story. Larry Summers is the president of Harvard University. Now you all know what we're talking about. Is there anybody who doesn't know? Still need to explain. Larry Summers addressed a meeting of economists and focused on development of science and technology a week ago today. It was last Friday. And at this meeting, he addressed the question of still surprisingly the fact that even after all of our efforts, there's still not equity in the numbers of women in science, especially at the top positions. Women are still not well represented in the top academic, industrial positions in the world of science. Why is that? Well, Larry Summers suggested, he said, he's not sure about, everybody talks about discrimination, but he's not so sure about that. He said he offered 3 explanations of why there's still an inadequate presence of women in science, women in mathematics and science, and he listed them in order of importance. The first, he thought, most important, he thought, was the fact that women do not want to commit themselves to very demanding and grueling professions. And science is a demanding and grueling profession. The second, there's a lot of research that suggests that there might be innate differences between women and men and in scientific ability. And third, he was willing to throw in the possibility of discrimination. Well, Nancy Hopkins, who is responsible for the big report at MIT, got up and walked out of the meeting. That gesture was reported in newspapers around the world. And in fact, just while I was waiting for Terry to pick me up, I was listening to the news on television, and there was a news broadcast about Larry Summers and the whole incident at Harvard. Well, there has been a huge uproar, and all through the week it has been growing and growing until finally Larry Summers was obliged to publicly apologize. And apologize. And apologize. However, he didn't say he was wrong. He just said it was thoughtless of him to speak his mind so openly, given his responsibilities as president. Now, I'm actually taking great pleasure in this whole event because it's opening up. It just opens things up in a way that's quite interesting. On this news program I heard before I came in, the whole phenomenon was turned around, and you all cheered when I said he apologized, and there was genuine outrage across the campus. And I have to tell you, the other thing that's important to know here is since Larry Summers took over the presidency of Harvard, the proportion of women hires, senior women hires, has dropped precipitously. So, or tenured, the tenuring of women. Last year, there were 32 male professors tenured and 4 women. So it's gone down quite a bit. So it's the conjunction of these two, his claims okay, but what happened on the television tonight is that it was turned into a story about political correctness, about the violation of freedom of speech. What happened to Larry Summers' freedom of speech? And Christina Summers got up and said, this is an outrage. He shouldn't apologize. He is caving in to PC, the pressures of PC. Well, I have something to say about this, actually. Let me try to bracket the question of whether, there is the question that a lot of people have a lot of interest in, to try to factor out the differences, the relative roles of nature and nurture in the development of behavior. I have to say, scientifically, this is one of the most meaningless endeavors I have ever seen. There is no way to parse the roles of nature and nurture in human behavior. There are ways, and especially there is no way to parse the roles of nature and nurture in human behavior because we are such social individuals. Let me just make 2 arguments. Let me step back. First of all, I have to point out, I just finished telling you there is no such thing as a gene except in the context. So already, what's nature? Well, a lot of people say nature is your genes. Nature is your DNA. But DNA doesn't do anything. DNA only acquires meaning from its environment. That the cell is the environment that gives meaning to the DNA. The intercellular structure is the environment of the cell. The organism is the environment for the tissues. The family is environment or the womb is the environment for the fetus. The family is the environment for the individual. Environment, there is a constant slide in where you draw the line between biological and environment, biology and environment, or between nature and nurture, is always a relative issue. It's always, there is no such thing as saying this is nature and that's nurture. There's no way of doing that. That's true for everything, for all of biology. There are particular problems with studying human behavior because of the importance of our human interactions to each other. So it is well known, for example, that people respond differently to each other according to the way they look. So it's known, for example, that tall men are more successful, have higher salaries than short men. They're more successful in the world. They're more successful in politics. Why is that? Do we think it's because there is a linkage, genetic linkage between the genes that are responsible, that influence the height of an individual and the performance in the business world? No, it's because people respond to tall people differently than they respond to short people. People respond to beautiful people differently than they respond to homely people. There was a study of the ratings of professors, their effectiveness as teachers. And guess what? Good-looking professors were better teachers. Isn't that amazing? People respond to individuals, if they identify them as male or female, differently. Now, there's nothing universal about this. We respond to each other according to our expectations. There may be cultures that think short is better than tall and would react differently. There may be cultures, in fact, I know there are cultures, I'll tell you a story. There was an interview with a leading Japanese executive, this was about 15 years ago, by a journalist, and he was asked, why is it there are so few women in top executive positions in Japan? And he said, oh, it's obvious. It's because women are so competitive. They don't know how to cooperate. So, how our behavior responds to what we see when we look at an individual depends on our culture. It depends on our beliefs. So when Larry Summers gets up and says there may be innate differences between the ability of males and females, whatever the numbers are, people are going to impose expectations on those girls and boys, on those women and men. That this is not a question of free speech, this is a question of responsible speech. That what Larry Summers says has consequences. It has consequences not only for the promotion of these women, but for their performance. Girls, in fact, do historically, or in our culture, girls have done less well at mathematics than boys have. Surprise, surprise. People behave according to our expectations of them. That human beings respond much more powerfully. We're extraordinarily plastic, adaptive individuals. Our biology that's what our biology gives us. our biology gives us our adaptability. And our adaptability means that we are going to shape ourselves. Our development is going to be shaped in response to our environment, in response to the expectations. So, yes, Larry Summers needed to more than apologize. I mean, if this is really a question, I have a responsible speech. So that's what I have to say about Larry Summers. Okay, yes.
To continue with another archaic subject, but unfortunately it's very much in the forefront in middle school and high school biology classes these days, is the idea that you talked about so many wonderful futuristic ideas and, you know, putting this together and putting that together and going forward. Unfortunately, in the classroom, we have this hand holding teachers back, saying, no, you have to teach intelligent design. You have to teach, I mean, it's kind of like saying, maybe in geology you should be teaching about flat earth theory. The thing that concerns me is that the academic communities, although I guess not in Harvard, is seemingly ignoring the fact that you have these wonderful kids growing up and going to classes and now being subject to having to make a religious versus scientific decision in the classroom, teachers being forced to teach things that don't belong in science. And what is the academic community, do they look back? Do they see what's going on? And what can they do about it?
Are there pressures on you in Oregon?
Oh, yeah.
Yes?
Very much so, yeah. My son, one of the teachers, luckily he did not end up having him for science, but he was having this giant debate about creationism versus evolution in his class.
It's very disturbing. I mean, I share your concerns. There's no question, and it's it's a very difficult time we're living in. The political sources of this current resurgence of creationism are obvious and don't need to be said. But I actually have some very serious criticisms to make of the scientific establishment here, not for their inaction so much as for their impoverished reaction. That is to say, I mean, I actually think it's so bad that there are now, we could say there are two kinds of fundamentalism at work. There's neo-Darwinian fundamentalism versus creationist religious fundamentalism. That the standard defense, the standard response to the threat of creationism is to dig in our heels and to say, insist that there is no problem with Darwinian theory. There are no gaps in the theory. I think that is a politically very foolish stance to take because there are gaps. There are problems, and those problems need to be addressed. And I think until the scientific community is willing to recognize the problems with evolutionary theory and to open up the conditions for scientific and for intelligent science, that we won't have we won't really be effective in our challenging of intelligent design. So that's my.
Dr. Keller.
Yes.
I enjoyed your lecture, and I believe the quality of your presentation, at least in part, makes up for the disparity in numbers of gender.
Oh, well, thank you.
I hope that I can phrase my question correctly the first time. Your lecture emphasizes the complex processes that occur past DNA
such that cellular processes take place. And given those complex processes, I wonder how you view the value of the Human Genome Project at this point. Do you think it was a worthwhile undertaking? Do you think it will ultimately be central to the understanding of cellular biology, or just a very meager first step?
I think it's very important. I think that the importance of the Genome Project has, the project has totally changed direction now. Once the sequence information was sufficiently reliably established, the focus is turned entirely to what is sometimes called functional genomics. The great value of the Genome Project is that sequence information is giving us tools. This sequence information is not telling us who we are, but it's giving us tools it's giving us probes for revealing the complex dynamics that the assumption of the initial assumptions of the Genome Project completely ignored. So that's the best thing about science, is that it moves in ways, it grows and develops in ways that can often be that are often unexpected and even can be counter to the initial assumptions. So the use of sequence information, the value of sequence information is undeniable. The use of that information to probe the cellular dynamics is a wonderful, wonderful opportunity.
Thank you.
I can't see it, but yes.
Yes, I'm a senior high school at Wilson High School here in Portland. In my ecology class, we've been studying systems dynamics, which very much applies to what you're saying. The creator of Stella said that no theory is accurate. You say that, meaning directly, it is strictly theoretical. You say that systems biology will allow the bridging between theoretical and experimental, but modeling, as you're saying, which is a great tool, I will agree, but how does it bridge that gap to experimental?
Models in biology, successful models in biology, are representations. They're simultaneously representations of the data and guides to the collection, to the performance of particular experiments. So when people construct models in biology, the best modeling is they are going back and forth all the time between a picture that they're trying to draw and the tools and probing that experimental system and using the information that comes back to refine the picture and using the picture to refine the probes. And it's a very intimate back and forth between theory and experiment of a form that I think you often do see in physics, but that's not the way we have been trained to think about physics. We've been trained to think about, institutionally, physics has been, since the beginning of the 20th century, divided into two disciplines, theoretical physics and experimental physics. So the theoretical physicists make the model, and then they go away, and they leave the experimentalists to do the check and to do the verification. And so it all rests, it's not this kind of intimate back and forth that you see in good modeling in biology.
Thank you.
We'll take these last two questions, just one there and one there.
Okay.
My question is that what do you expect to do when you finish all of this stuff and you have all of this mapped out? You know how everything works together. I know that 60-some-odd% of the world is a quote-unquote creator-based religion. So what do you expect to tell them about how you're going to start messing around with genes? It's kind of my question.
I'm not sure I understand the question. I hear two questions, so help me out here. a question that I hear is, what will you do when you have all this information and you've figured it all out? Is that one of your questions?
Yeah, and then the other one is like, what's going to be your response to the religious community?
Okay. I don't think that we're going to figure it all out.
You don't?
No. No. I don't think there's any reason in the world to expect that we can understand the universe. Why? We're part of the universe. I don't think there's any reason in the world to expect that we will be able to understand our own brains. How are we going about this? Our brains are products of evolution. They are shaped for surviving in the world, they've given us, our brains are very good. They allow us to do lots of things that increase our longevity and all kinds of things. But the one thing that brains were not evolved for is to make a theory of the brain. I mean, why would one expect the very finite biological capacity of the human brain to be able to encompass everything around it, be able to encompass the very biology of which it is a product? So I don't think we're ever going to get it all. What do I say to my response to the creationists? I think the problem of the apparent design of living organisms needs to be addressed head on. I think we need to think much more creatively about the nature of biological organization and how that comes about in order to provide a plausible response to the creationists. Michael Behe there's lots wrong with his arguments, but the question is real. The question is real. It's very difficult to make sense, to give a persuasive neo-Darwinian explanation for the evolution of really complex biological traits. We've got to do better.
Thank you.
Yeah.
Yes, Dr. Keller, thank you for coming to Portland. You talked about the shift from the analytical approach to the synthetic approach in genetics. Are you seeing that same kind of shift in other disciplines? Is it perhaps a general change in society's way of thinking, do you think?
Oh, that's an interesting question. I am seeing it in other disciplines. I have to think about that. I don't have an answer. I'm sorry, but thank you for the question.
Okay, you bet.
Okay, thank you. Stage, otherwise people will
All right. Yes.
I just want to comment on something you said in the question and answer thing regarding Larry Summers and politically correct speech.
Yeah.
I think your comment about politically correct versus responsible speech was right on, because I think there are enough people who are, if you will, undereducated, and those kinds of comments do extreme damage because people who are undereducated believe them.
Oh, absolutely. So thank you. Yeah, no, I think that's right. Yeah.
Okay, so I read bestsellers, including Michael Crichton and Prey, which I assume will be a film. Is it going to be a film? Anyone know? Okay. Crichton? Yeah, it will be a film. Okay. So it's got little nanobots that swarm and fly around, and they basically wreak havoc and kill people. Yeah. So my question is, you've talked a lot about the convergence of a couple metaphoric systems, one being to show the circuit boards and the computer science, the machine thing.
Right.
And biology over on the other side.
Right.
So of course we all wonder, the Frankenstein sort of question is, when do we get machines that we can't really tell apart from biological systems? It becomes an academic question whether they're alive or not. And maybe we don't even care. So do you see that coming along? Coming along nicely?
No, not nicely. It's a question, I actually have argued that, okay, let me back up a little bit. The question of whether, whether a robot or an X is alive or not assumes that there is some definite category of, there is some essential defining feature of life. And it assumes a question, or it presupposes a question that is very popular in some quarters, not others. And that question is, what is life? Now, that's a very peculiar question, what is life? Because what it does is leave out the question, life of what? What is life? Are we talking about a cell, or an organism, or a tissue? I mean, the life of what? That question comes into play with the assumption that there is some essential property called life. I don't believe that, I don't even know what that means. I think that there are living organisms are very complex and organized in very really interesting ways. I think that there are other kinds of structures that are organized in really very interesting ways, like the internet or robots. Where we draw the line, I think that's a conventional question. I think it's a historical question. Historically, there was a time when were viruses alive or not alive? Where are we going to put them? Plants. Well, they weren't quite, in the whole, we have a word for living, for vital, it's taken to be synonymous, animate. Animate, as a description of life, includes plants. Animate means motile. For me? So, historically, plants were sort of alive, but not quite alive. It was only in 1800 that plants and animals got pushed together into a single category of living beings. It's only in the 1930s, 40s that we decided viruses were alive. Even bacteria weren't, it wasn't clear that they were organisms. They were ambiguous. So I think that I don't think there's a sharp dividing line between kinds of organization that we recognize in conventional organisms and other, mechanical things. I think that there are all kinds of gray areas, and robots undoubtedly will approach that gray area. And then it will be up to us whether we consider them alive or not. That's my view. Yeah, Rebecca.
Hey, you actually talked in your speech about how certain genes were not alive, and this is kind of on the same branch. How do you define what is a living gene and what is not?
Well, I didn't say there were some genes that were alive. I said the genome was not alive. It doesn't. That's not a gray area. Genomes by themselves exhibit none of the properties that we attribute to living organisms. They just don't do anything. They're like rocks. All right? And that's... Yeah.
Yeah, I'm an electrical engineer, and I have two simplistic questions, looking at it from my point of view. One is modeling. How do we model that? Can you take a DNA string and model it so that you can figure out what's going to happen? And can you turn that into a model too under experimental biology? Is it possible to take a string of DNA in the lab and a chemical and see what happens? See what proteins it
You
makes?
put a string of DNA in a test tube, and nothing happens.
Yeah.
DNA is very inert. By itself, nothing happens.
So what is experimental biology?
experimental biology is experiments usually with organisms or subsets or parts of organisms, parts of cells, but it's never. But it is true that DNA by itself doesn't do anything. So, yeah, okay, yeah.
That's the reason for my question about microchips.
So I would say good luck. Okay. This relates to a question that came up before. Most of what goes on in the brain has nothing to do with gene expression. Microarrays or gene chips are ways of recording the activities of different genes. So you might have 100 different genes that you're or sections of the DNA that you're trying to track, and you have probes that will measure the amount of messenger RNA that is being produced. It's the and it's this shift from looking at genes to gene expression is extremely important. So for example, the original Human Genome Initiative was sold to Congress on the promise of curing disease, right? It was going to, right. And now, today, it's universally acknowledged that the key to disease is not in which genes you have or what the sequence is, but what are the patterns of gene expression. So microarrays or gene chips can give you the patterns of gene expression, and the idea is to develop, to fine-tune the medication to the specific, very individual patterns of gene expression. Most of brain processes do not involve gene expression. Most brain processes are just about the dynamics of neural processing, of neural firing. Genes get activated. I mean, if you look at the work on memory, what's his name? Eric Kandel's work on memory. A short-term memory does not involve gene expression. Only long-term memory involves gene expression. So we don't know about, I wouldn't.
But couldn't these have to do with receptor sites or enzymes or?
Sure, there are receptor sites and enzymes, but there's a lot of stuff. I mean, that's all operating in the brain and as in the rest of the body.
Could it possibly show us an aberrant receptor site?
Yeah, it could have shown, but we can have aberrant receptor. Yes, we can have aberrant receptor sites that might be aberrant because of a genetic mutation. That's true. Or,
that would tell us a great deal.
but it also is true that there are aberrations that are caused, and maybe even aberrant receptor sites because of faulty regulatory systems. One of the big sort of developments in recent years, in the last 5 years or so, is the discovery of the importance of microRNA molecules and regulation. That these are little tiny RNA molecules that maybe they came from the DNA, maybe they came from somewhere else, but once they're there, they just take over the system.
So we're not going to see them on a microchip then?
Oh, you will see their effects on the microchip. You will see their effects, but you won't necessarily see their genesis.
You're not very encouraging.
For a unified theory of mental illness, no.
Thanks, just a second.
Maybe I will take another cup of decaf if they.
Keep you awake.
Yeah.
I'll shift gears a little bit from scientific detail. The lady that asked the creation versus evolution question, I think she stormed away from the microphone.
You did?
When you said that, perhaps the Darwinist is as dogmatic as the creationist, and you were encouraging some debate.
Yeah. Not between the two fundamentalists. I think debate between fundamentalists is absolutely useless.
That's what I was going to say. In what grounds would you encourage such a debate? And is there a place where you've laid out some framework in which this debate might be held.
I don't know that debate is the right response. I think that, what I would like to see is, the diehard reds versus the diehard blues. Forget them. It's the middle ground that we're concerned about. That I think we need to respond with, just provoking, intelligent, and serious scientific reflection among people who are not fundamentalists about anything, about these issues. I'm rambling. That's the effect of that second glass of wine. Here's another sip of coffee. Yeah.
So you indicated that you thought that a lot of biology is being driven by the marketplace at this point,
Yeah.
with pharmacology and the like.
Especially in biomedicine, yeah.
Yeah, with biomedicine in there.
Right.
And it's curious to me. I can see why you think that's a problem. I'm on the other end of that as a cancer survivor, and I'm really interested that they are trying to find these kinds of things. So I'm wondering about what you see is the pitfalls of having a science that's being generated in that way.
Well, I don't, with all due respect, I would question whether your survival has much, if anything, to do with the medications, the chemotherapy, whatever you were given. It's wonderful that you survived, but the assumption that it's because of medical developments, I think, is a very shaky assumption. It is interesting. There are now, for the very first time, there are now drugs that are being put on the market that seem to have, cancer drugs that seem to have some effectiveness. It's scandalous how damaging chemotherapy is, and how little benefit there is. So, it's just a huge scandal. So what's wrong with market-driven? Well, that's one thing that's wrong with market-driven. We're all so dosed up on all these drugs, and what are they doing to us?
But isn't that if it really is market-driven, is it just that they're being, it's in the development of it, but not the follow-up or the understanding of what it's doing?
Oh, the problem with market-driven biomedicine, or particularly around pharmaceutics, is that first of all, we don't know what's effective. We've really lost control over measurements of effectiveness, and we also. It has been argued that there have been very few new novel developments in drugs in the last 30 years. That what has been, is just little tinkering in order to get new patents.
But is there any driving on the other side of it, that it's looking not so much at the pharmaceuticals or how you cure, once it's there, but anything in terms of just understanding the mechanisms that cause these various diseases, and if there's ways to, you
there
understand
is
sort of where I'm going.
Yeah.
Is that kind of research, or is it all going into just let's get better and better drugs?
Or let's get more and more. I'm not even sure that we're getting better and better
Well,
drugs.
let's get more drugs that cost more.
That's right. That's the problem.
Anti-cancer drugs that are $300 a buck.
Yeah. No, there is. It's not completely market-driven, thank God. there is some effort, but the market forces are really working against that. So, for example, I have to tell you that my book, The Century of the Gene, was savaged. And it's a good book. There's nothing in that book that is not scientifically defensible. And so I asked a lot of biologists. I was stunned by the response, and I said, well, "What's going on?" And they all said, "Money." And in fact, I don't know if that's true or not about my book, but there was an exhibit at the American Museum of Natural History that was, there's a lot of hype about genes and what they do, the power of genes, and this was one of the most egregious exhibits. The hype was outrageous. What genes are, particularly the relation between genes and diseases. Where did that exhibit come from? Well, it turned out a group of biotech executives had convened and decided the gene needed some promoting. So they provided the funds for this exhibit on the condition that the source remained anonymous, and then it got leaked. That's how I know about it. But so that's counter to the effort to find out what the mechanisms really are.
Maybe one more question.
Okay, go for it.
Just the way you're explaining the genome, that basically we peeled the onion and we found it's not as simple as we thought it was, and we've got all these interactions.
We peeled the onion and we got to the core, and the core is useless. I mean, it's not useless, but it's not giving us the onion.
Yeah,
So we have to put the layers back, right?
But the question, this is a self-replicating system that has very complex interactions, the way it works. Is anybody working on how did it evolve? I mean,
Of course, yes, but it's not. You're talking about the origin of life.
Yeah.
Well, the dominant explanation is that there are RNA molecules that can catalyze their own. They can catalyze their own reactions. That's the dominant view. It's not my view. The other semi-dominant view, prevalent view, is symbiosis, that there were independently evolving autocatalytic systems, and then they were evolving some kind of molecules that could serve as memory devices, and they got together, and voila. You put together two very different kinds of things, and you get something new, like a cell.
There's the answer to the origin of life, and I say thank you.
Well, thank you.